for giving me this opportunity to present the lead paper on exploring the endophytic Bacillus species for the induction of innate immune response. So here, I would like to narrate what is our journey in relation to the induction of implantar resistance for the management. So how the endomicrobiome can be engineered for the management of fungal, bacterial, and viral pathogens. So in this case, these endophytes, they trigger the implantar resistance by suppressing bacterial, viral disease, and fungal disease. But how that this happens? Hello. I think there is some interruptions is coming. Okay, so in this case, so how to discover? So what are the endomicrobiome which is found to be available? So where they are located, and this in this case, our journey was started right from late blade of potato. So which was started from Sikkim and to Nilgiris. So the pathogen, which is a necrotrophic pathogen, Phytophthora infestans, was responsible for causing this late blade of potato, which was isolated from the latching area of Sikkim, and these are the symptoms which is associated with the late blade of potato. So for the management of this, we framed a hypothesis whether the endomicrobiome confer the resistance to Phytophthora infestans or not. So this was the one which was hypothecated, and based on this hypothesis, the pathogen was isolated from latching demong. And after the isolation, the pathogen, we have to necessarily go for confirmation based on the hyaline hyphae, based on the lemon-shaped sporangia, based on the semi-papillate sporangia, internal transcript spaces, RXLR, AVR3A, beta-tubulin, G protein, beta subunit, and 1,3-beta-glucanase. So these are both the phenotypic and molecular based identification was carried out. And this is the phenotypic character for the aseptate mycelium with the Phytophthora, with the papilla, and all those things. And this is the one where the electron microscopic observation of the pathogen was isolated, re-inoculated for confirming the pathogenicity. And this is the Phytophthora which you have observed. And here this Phytophthora results in the release of the zoospores, and it results in complete release. So based on which we confirm the pathogen associated is Phytophthora. Then apart from that, this Phytophthora zoospores, they come in contact with the stomatal opening, and the germ tubes come and grows through that of the stomatal opening and causes the infection. So if it is the case, does endoscopy engineering with a bacterial antagonist mitigate late blade of potato? So that was the question which was brought in front of us. So for addressing this particular issue, the endomicrobiome and then the rhizomicrobiome was engineered for effective antagonistic bacteria, so that we prime the resistance mechanism associated with the plant for sustainable management. So the microbe associated molecular pattern triggered immunity was found to be explored by the application of this endomicrobiome, so that it confer resistance to late blade of potato. So to understand the endomicrobiome diversity, so the various solanaceous crops and potato was isolated from different areas of India, starting from Sikkim, Andhra, then Tamil Nadu, and in different places, including Karnataka. So out of that, we have isolated 644 bacterial endophytes was isolated from different types of the plant pods, starting from flowers, leaves, stem, then root and tubers. So these are the cultures which was maintained. So if we analyze what is the diversity profile of these particular bacterial endophytes associated with the potato and tomato and other solanaceous crops, so we were able to come across with a new bacterial endophytes like Spingobacterium thalphyllum, then Pseudomonas geniculata. Then from the stem regions, we were able to isolate Pseudomonas geniculata, then Serratia marcescens, Bacillus willisensis, Bacillus subtilis subspecies speciesensis, Bacillus subtilis subspecies stercoris, and Spingobacterium chango-NC. So like that, the diversity was going on increasing based on the analysis, where they were all grouped into 3 different groups of this particular bacterial endophytes. So if it is the case, what is the antagonistic potential of the bacterial antagonist that is against late blade of potato? So these were found to be screened. So the bioefficacy, so this is in this particular picture. So we can see how the Phytophthora completely colonized the untreated plate, and this is where the complete suppression of this Phytophthora is found to be noticed. So all the 600 isolates has been screened for its efficacy in selection only. So few slides I have focused here. So this is a Serratia which is having an inhibition, and here this Phytophthora has been completely inhibited over this particular case. And here there's a complete inhibition. So why this inhibition? What is the biomolecule which is found to be associated in relation to this type of inhibition? So based on that, if we assume, so the most of the bacteria which was responsible for producing the biomolecule and suppressing the plant pathogen Phytophthora infestans was identified as 80%, which comes from the firmicutes, and rest of them 12% comes from proteobacteria and 10% comes from bacteroides. So if so, the endomicrobiome, when it inhibits, whether it inhibits the late beta potato or not. So that was the question. So here this inhibition is because of the production of antimicrobial biomolecules during the interaction of the bacteria with that of the fungal pathogen. So here in this case, the biomolecules like streptozotocin, then ethanamide, clindamycin, 3-deoxy-D-manoic lactone, phosphothiolic acid, and glucuronamide. So these are the different types of the biomolecules which are found to be identified from Bacillus safensis, which is an endophyte which is responsible. So though these organisms produce these biomolecules, so does they have a chance to produce a biofilm so that it goes and binds with it of the target area or the niche where the Phytophthora is found to be associated? So most of the isolates, out of the 640 isolates, we narrow down those isolates to 10, which has more production of the biofilm and due to the production of the lipo extracellular polysaccharides. And the diversity of the antimicrobial peptide genes were also assessed in all these effective bacteria, where most of the bacteria which we have selected out of the 10 from the 640 isolates, they were having different types of the antimicrobial peptides, which is accounting for around 13 different types of the antimicrobial peptides has been detected. And here, since it has such a potential, we try to engineer the phylloplane with that of the endophytes. So when we apply, so this photograph indicate the colonization of the potato leaves on the leaf surface by the Bacillus willisensis, which has been colonized. So such a huge colonization has been noticed due to the presence of this. And when it comes in interaction, so this is one of the interesting scanning electron microscopic photograph which explain the sporangia is found to be now this is colonized by the Bacillus, the amyloliquefaciens isolate 345. So all the bacteria, they move towards the Phytophthora sporangia and they go and colonize. And not only the sporangial colonization, it also colonizes the hypha, which is found to be intermingled with that of the sporangia. So this results in cleaving of the sporangial wall. So after this cleaving of the sporangial wall, the bacteria multiplies inside and subsequently it also colonizes the hyphal region. So this is the hypha where there is a complete colonization of this particular bacteria, and thereby it cleaves the cell surface and of the Phytophthora and it leads to lysis. So likewise, it was also recorded in Bacillus willisensis isolate 261, and the most effective bacterial endophytes we have identified based on the molecular fingerprinting as Bacillus amyloliquefaciens, Bacillus willisensis, then Bacillus subtilis subspecies speciesini, Bacillus subtilis subspecies subtilis, Bacillus safensis, then Bacillus subtilis subspecies stercoris, and Bacillus subtilis subspecies subtilis. And these were the effective organisms which we have documented from our studies. So after that, as the chairman in his introduction, whether these products are found to be made available, the biomolecules in his remarks. So this particular one, it was developed into an formulation of the aqueous formulations was developed, and this aqueous formulation, when it was applied on the leaf surface, how do they interact with the other plant during the tritrophic interaction, how the interaction takes place. So during the time of the tritrophic interaction, if you take, in the presence of the pathogen alone, if you take, so these three different compounds, that is the compound number methyl adipic acid and other three compounds was found to be produced by the Phytophthora when it is found to be present in that of the leaf surface. But in the presence of the Bacillus altitudinus and pathogen, so here the particular compound which is found to be produced when pathogen alone was present on the leaf, so that particular compound was totally absent in this particular condition, which indicate that this particular endophyte has the capability to break down that particular molecule, or it has prevented the production of that particular molecule which may be responsible for inhibition. So this is the other case of the Bacillus subtilis, and that is Bacillus safensis, which is another interesting bacterial endophyte which has also resulted in the non-expression of the particular compound responsible for the pathogenicity, whereas here it has produced a compound in addition where hen tricontain and 5-hydroxymethyl furfural. So these two compounds were found to be produced in addition to that of the pathogen compound that was as a biomolecule produced. So this is how they are found to be different. So likewise, we have documented these for different effective bacterial endophytes. 3 different bacterial endophytes has been documented, where that is a Pseudomonas geniculata. So this is another one, because recent days, if you see, Pseudomonas fluorescens also has become as a human pathogen. Pseudomonas aeruginosa is also found to be a human pathogen. And Pseudomonas fluorescens has been reported as a human pathogen in 2014, where it becomes as a spinal cord fluid infection contaminant. So Pseudomonas fluorescens cannot be used, Pseudomonas aeruginosa cannot be used. So in that case, a non-pathogenic Pseudomonas, which is Pseudomonas geniculata, can be used, where this is also found to be as responsible for the suppression of the biomolecule produced by the pathogen, which is responsible for causing pathogenicity reaction. So that has been reduced here. And so that is next is the Staphylococcus thalphophilum. So that was also found to be another interesting organism, which is of a new one, which can be used for the management of the late blade of potato. So this is the case where we have studied. And so ultimately, if you put all the cycles put together, so this is a KEGMAP pathway has been analyzed. So when these different types of bacteria has been used for identification, so they produce different types, this green colored compound, fructose 6P, then acetyl carnitine, ethylene, next fumarate, succinate, then hexadecanoic acid, octadiene, alpha linoleic acid, dodecanoic acid, traumatic acid, traumatin, and gibberellic acid. So these different types of hormones and the biomolecules has been produced during the tritrophic interaction. So during the tritrophic interaction, if they are producing these molecules, they are involved in different types of the pathway. That is TCA cycle is there, electron transport chain, oxidative phosphorylation, citric acid cycle, ethylene pathway, aspartate pathway, terpenoid backbone pathway, glycolysis, beta oxidation, then fatty acid biosynthesis. So these are the different types of various pathways which are found to be associated with the defense mechanism to trigger the immune response of the plant and thereby result in the suppression of the plant pathogenic establishment on the leaf surface. So the tritrophic interaction by the bacterial endophyte on the phylloplane produce these biomolecules in different pathways and results in the suppression. In addition to that, we were also going for the detection of the presence of different types of pathogenesis related genes. Defense related genes were also found to be documented, where it is too early for us to disclose those results. So now, based on this, we came to a conclusion that this particular application of this bacterial endophyte play a vital role for suppression of the late bed of potato pathogen on the phylloplane, and the establishment has been blocked. So not only that, so next we went for the investigation. What might be the role of these particular bacterial endophytes on the suppression of the viral diseases? So that was the another one which was also found to be our interest. So in most of the cases, viral disease, till my student life, till now, it is cut and burn is the technology which is found to be available. So here we attempted to innate immunity, to trigger the innate immune response, which is mediated through LR virus. So this is a tobacco streak virus in cotton. So we isolated all the bacterial endophytes from that of the resistant genotypes. So these are the diversity of the bacterial which is found to be associated with that of the resistant. The green color one are the bacteria, Bacillus cereus, licheniformis, subtilis, amelifications, and endophyticus. So all these green colored bacteria, so these are the one which is isolated from the healthy are the resistant genotypes, and these one are the infected from the infected plant, Bacillus cereus and velicensis and the Enterobacter. So these are the different one which we have isolated. So here what is the thing is the diversity profile. If you see, the diversity is found to be very high in the case of the healthy plant and in the resistant genotype, which is found to be playing a vital role for suppressing the expression pattern of the viral infection. So based on that, so this particular paper, it was published, we have published this in Frontiers in Microbiology. So this particular work has been already published one. So here if you see, when we apply this Bacillus amelifications VB7, when you challenge it and through simultaneous inoculation with that of the virus, so here there is 100% reduction in the lesion was found to be noticed. Whereas in inoculated control, more than 230 lesions was found to be observed due to the suppression of the viral infection. So here, this particular one indicate that, so when we apply this particular bacterial on the leaf surface, so it resulted in the reduction in the number of lesions in the case of Bacillus amelifications and Bacillus licheniformis. So these two bacteria reduce the viral lesions to a greater extent when it is applied through simultaneous application. So likewise, the same thing, it was also applied under field conditions also, where the Bacillus amelocubations, when it was applied in the cotton hybrid RCH659, which was found to be susceptible for the tobacco streak virus, where the percent TSV incidence, the tobacco streak virus incidence was around 22%, whereas in untreated control, it was around 45%. So there was a 50% reduction in the virus establishment, and this indicate that the efficiency and the plant growth promotion was also, the plant height was found to be increased by the application, and besides that the variation in the virus titer was also observed. So the virus titer was found to be very much reduced in this particular case through ELISA reading. The virus titer was 0.5, whereas in inoculated control or in the control it was around 2.5. So 200% to 300% increase in the virus titer was found to be observed in this particular case, and it was conducted in the field trial was conducted for this, and this is the hypothetical model which explains that this particular bacteria, Bacillus amelocubations or the Bacillus licheniformis, when we apply, it results in suppression of the viral disease. So this particular paper we have published in Agriculture, Ecosystem and Environment during 2018. So not only that, we were also testing this particular effect on chow chow, where the chow chow Asian mosaic virus we are trying here. So the Bacillus amelocubations, when the chow chow planting material or the chayote, when it was soaked in the bacterial suspension of Bacillus amelocubations at 0.5% for 20 minutes, it resulted in complete suppression of the virus expression on the disease symptom under the field conditions. So this is the one untreated control where we can see the symptoms, and whereas in the case of the Bacillus amelocubations combined with the root extracts of the Mirabilis jalapa, the disease incidence was found to be very much reduced, and the Bacillus amelocubations 0.5% and the foliar spray of the root extract of the Mirabilis jalapa, it reduced the percent disease incidence to 1%, whereas in untreated control it was around 83%. Infection was found to be noticed with an increased yield or supplemented with increased yield. If so, why the suppression? What is the reason behind the suppression? So to understand the mechanism, we went for the whole genome sequencing of this particular effective isolate, namely the Bacillus amelocubations. So this is the whole genome sequence data, where the data reflect that based on the 16S rRNA, the bacteria was identified as Bacillus amelocubations. When you go for whole genome sequencing, it was identified as Bacillus willisensis through Illumina HiSeq platform, and the genome annotation was done through RAST tool, and the contigs was 1 contig, and the genome length was 3 MB base pair, and the protein coding sequences was 3090 coding sequence, and the hypothetical proteins 506 hypothetical protein, and the functional proteins are found to be 2584 with 8 secondary metabolite clusters which was found to be noticed. And this is the whole genome sequence. When it was compared, it was identified as Bacillus willisensis. So the genome annotation, when we go, so it reflected the presence of NRPS non-ribosomal polyketide synthase in region 1. Region 2 was corresponding to PKS polyketide synthase. Region 3 was terpene synthesis, and region 4 was NRPS trans ATPKS. Region 5 was trans ATPKS, and region 6 was NRPS cluster, which is coding for bacteriocin. Then the cluster lantipeptide, which is a very potential antimicrobial peptide, which codes for deficidin of antiviral in nature. And this totally, if you see, so these 14 secondary metabolite clusters were identified, which are responsible for synthesis of surfactant, bacillibactin, bacillicin, deficidin, fengicin, bacilliin, macrolactin, calimantin, butyrosin, and plantathiazolin. So these were the things. So when this particular bacteria, which is having this type of potential antimicrobial peptide with the different clusters, we went for the tritrophic interaction. So how they are going to trigger the MAMP triggered immune response. So the Bacillus amelocubations and the simultaneous inoculation here in the localization host, the lesion was completely suppressed compared to that of the inoculated control. So this is the Bacillus amelocubations and inoculated control, where the symptom reduction has been observed. In Bacillus licheniformis also, here we can see how the reduction in the number of lesions has been noticed. So if so, what is the mechanism of action involved in suppression? So the reason is behind here is microbe-associated molecular pattern triggered immune response has been noticed. So the molecules which are found to be associated for triggering the immune response are flagellin, elongation factor, peptidoglycan, lipopolysaccharide, swellanine, cell wall beta-glucan, and cell wall chitin. So these are the non-self molecules of the plant of bacterial origin responsible for triggering MAM-triggered immunity. So if it is so, is there any regulation of the different genes is the next question that we arise here. So what is the impact of suppression on GBNV if these different genes are going to be regulated? So for which the clones, agroinfectious clones expressing this gene of elongation factor and flagellin was developed. It was applied through root drench method, and after the application, we went for assessing the various defense related genes and the transcript level of mitogen activated protein kinase, then the Ricky factors, TGA, then MyB, MyC, then PAL, ERF, PPO, and this and proline, superoxide dismutase catalase, and all these defense genes were found to be detected through real-time PCR assay. So the real-time PCR based on the gene transcript level expression, it explained that the mitogen activated protein kinase gene expression was found to be triggered. For example, there was a down regulation in the case of the control, where inoculated control there's a down regulation, whereas with respect to 0, 24 hours, 48, 72, and 90, up to 120, after 5 days, the gene expression was detected. So the study indicated that the expression pattern of the defense-related transcript genes were found to be more in the case of the bacterial cells of Bacillus applied on the leaf surface. Whereas in the case of the same trend was also observed in the case of the gene which was applied, that is the agroinfectious clone, which has been applied here on at which is 1.58 and 1.99. Whereas in rest of the cases, in the treatment like BTH and it's a bion, then agrobacterium as such, whether it is able to trigger the immune response or not. So they were found to be having a very limited expression. But here what we'd like to conclude here is the flagellin and elongation factor trigger the transcript level expression in this particular case of mitogen activated protein kinase. And similarly, the Ricky factor was also found to be enhanced in this particular case, followed by the NPR1 gene, which is responsible as a hub for triggering ISR and SIRA was also found to be triggered in this case. And later, if you see, the PR1 gene expression was also found to be induced, where the level was found to be increasing up to 3 days. Then if there is a slow decline was found to be noticed. Whereas in the case of the inoculated control, down regulation was found to be noticed here. And in case of PAL, the phenylalanine ammonia lyase, there also the increased expression has been noticed here. So likewise in polyphenol oxidase, the expression of the different gene expression was found to be more. And in the case of that is in the case of the lipoxygenase gene. So in the case of lipoxygenase, which is a very key enzyme, which is associated in triggering the ISR and SIR pathway. So there also, if you see in the case of Bacillus, the expression was more. And in the case of the elongation factor and flagellin also the expression was there. So up to 4 days, the expression level was found to be more. Then jasmonic acid response, the methyl jasmonate transcript levels were also found to be increased, followed by transcription factor MIC gene expression. Then PDF 1.2 gene expression was also found to be noticed. So here, when considering all these different gene expression, we came to a conclusion that all these might have contributed for the suppression of the virus associated with the reduction of the lesion. And we would like to substantiate that through ELISA for the virus titer, which indicate that in inoculated control, the virus titer was found to be 1.2, whereas in case of flagellin, elongation factor, and Bacillus and BTH, the virus titer was found to be more than 200-fold reduction was noticed. Not only with that, we also documented what is the level of the core protein CP gene expression. The CP gene expression was also found to be very much reduced, which was 10 to the power 5 in the case of inoculated control, whereas in Bacillus amyloicubations it was 10 power 2, whereas in case of flagellin elongation factor it was 10 power 3. And finally, we would like to conclude here is, so this particular application of this particular bacterial secondary metabolites or the agrobacterium which has the clones of elongation factor and tubulin, they trigger the mitogen activated protein kinase, then MAPK, then transcription factor, then followed by the PR1 gene in the nucleoplasm, it gets triggered, where it goes and triggers the JAR1 response, that is jasmonic acid, and also the NPR1 gene, which also goes and triggers the phenolic compounds and disease resistance, which is also associated with the SIR. So our concept where we say that bacteria or the biocontrol organisms, they are associated with induced systemic resistance. But now the induced systemic resistance is also activated and the SIR is also activated by the application of the biocontrol agent. And that is the one which has been proved by the different gene expression. So this is where the diversity of the metabolite has been triggered in case of VB7. When we apply, that is Bacillus willisensis, the cytosterol, then triaminocyclone acetonide, succinic acid has been noticed here. Whereas in inoculated control, the total metabolite profile was found to be totally different. And the validation of the effective consortium against the GBN and the field conditions also, this was found to be effective. So to conclude, so these are the different mechanisms that are found to be associated in the suppression of the virus, which is responsible for triggering the microbe-associated molecular pattern in the plant system, and thereby it is responsible for the suppression of the viral disease. So this is the hypothetical abstract which you have made based on the findings, which says that the virus disease has been suppressed because of this. So the message that I would like to give through this presentation is, so if we have a one better microbe in our hand, definitely the microbial product will grow at a faster rate based on the that is a recent survey which has been made by an international concern. So the investment in microbial discovery and development is going on increasing. So because the integration of the cost-effective alternative, everywhere these biocontrol organisms are going to be incorporated, and there is an improved market access for biopesticide and biostimulant, so which is responsible for going to increase the $11 billion in 2025. Whereas if you see out of the biopesticide which has been registered with Central Insecticide Board, Trichoderma viridi, Trichoderma hartianum, Pseudomonas fluorescens, and Bacillus subtilis. So these are the 4 organisms which is found to be present right now. But if you take Pseudomonas fluorescens, it is a human pathogen which cannot be advocated further. So only the 3 organisms are found to be available. So the message here is, so identification of new potential bioendophytes can pave a way for increasing the market share, and which can give an economical return to the farming community by the suppression of bacterial, fungal, and viral diseases. So as the chairman in his talk, he told that the availability to the farmer's end is found to be limited because there's a stringent registration norms enforced by CIB, for which definitely a public-private partnership is very, very essential to take these research findings to the farming community. Otherwise, all these findings will be in terms of an F-top tube in the -80 or -40 deep freezers, the bacterial cells will be sleeping. So this warrants a lead for a public-private partnership in a strong way. So with this, I would like to thank all my research team members over here. So once again, I thank the organizers for giving me this particular valid opportunity for sharing our journey on endophytes. Thank you, sir.
Congratulations, Professor, for the wonderful presentation. And I'm very much impressed by your electron micrograph photographs. That's very nice and self-explanatory. But I have a few doubts and say questions, but for want of time, I'm limiting to 2 or 3. Say others, say if they want to, they can also ask. Say one is Bacillus, your species is selective against infestans, or is it against all Phytophthora?
So we have tested only for Phytophthora infestans.
So if it is effective against, you can try on that Phytophthora other species, because Phytophthora, if it is Bacillus is effective, this is going to be one of the breakthrough in your research findings. That's one
Definitely.
thing. Then have you started your work on CAB registration?
So this particular project is, yes, sir. This particular project is an collaborative project which is funded by Coromandel. So the registration part is taken by the company.
If it is Coromandel, they'll be able to do it, because private agencies, they'll be able to do. But government agencies, yes, I think it's it's really difficult.
Yeah, definitely, sir. Definitely, sir. Because we face the same problem in our university also. So this is a funded project from Coromandel where they are taking it as an international patent.
Fine. So I got a few more questions, but I'll say there is no time left. So if our co-chair, if you got any doubt, can.
So,
Dr. Anagappan?
yes, sir. Yes, sir. So, I am observing the developments along with Dr. Nakiran. So, there is no doubt in this development of Bacillus against fungal and viral pathogens. So, the rest of the people can ask any query.
Sir, good morning, sir. Very wonderful presentation, sir. So I'm having doubt that.
Please identify you. Please identify.
Sir, I'm Dr. V. Bhuneshwari from Sri Shakti Institute of Engineering and Technology, sir.
Thank you, Bhuneshwari.
Okay.
I want to know whether this MAP kinase and MAP KK pathway genes also you have analyzed the signal transduction part to find out the signal transduction pathway. You have shown that MAP K and the.
Yes. What is your exact question, please?
That map, the pathway is involved. I want to know whether this, how this this MAP KK gene was increased, whether it was increased, and MAP KKK was increased or not, like that, sir. The expression
because mitogen activated protein kinase. So this is one which is highly essential for phosphorylation. And this particular MAP kinase, if it is getting increased, then naturally what happens here is, so the ISR related mechanism and the NPR1 gene expression will be simultaneously go and it will be triggered. So for which these transcription factors are highly essential. For in that angle only just we were investigating. So because if this NPR1 mediated gene expression has to be induced, naturally the transcription factor responsible for that should be also increased. So because of that we studied the one, and for that we came to a conclusion that this is also found to be playing a vital role based on our transcript analysis.
I think we conclude this session now because we don't have much time left. So I once again congratulate Professor for the wonderful presentation. And let's move on to the second paper.
That's, sir, hello.
Pardon me?
Excuse me, sir. I have just a small question. Nakiran, sir. Myself, Seshakiran.
Yeah.
Sir, like a few days back, we tried to isolate some antagonists from the soil, and it was exhibiting good broad spectral antifungal activity. And we sent it for identification. It came as a Burkholderia cepacia. And so I just have some, when I gone through the literature, it is in APS, it is giving a Burkholderia cepacia, it is causing even nosocomial infections in human beings. So what you suggest in this?
So better in that case, better it is not advisable to investigate further on that.
Okay, sir.
But for academic purpose, for publication purpose, there is no harm to work on that. But even if we can publish it, there is no issue in that, but it cannot be translated into a product.
Okay, sir. Even I feel some, sir, even they have registered in US as a biocontrol agent. So I wonder, like, when it is a...
Yeah, because what I'm saying is, so even if you go for the whole genome sequencing, the species even may get varied. And another thing that you'd like to say is, earlier we were saying that it is Pseudomonas fluorescens we recommended. But now Pseudomonas fluorescens in 2014, it has been stopped because it becomes as a human pathogen. So when there is an advancement in the science, the identification is also getting advanced. So based on that, naturally we have to accept certain things if it is found to be pathogenic to the human beings.
Say microbes are more dangerous than chemicals, because chemicals we can stop manufacturing, but microbes if you release to the atmosphere, that will be there. So we must be very, very careful while dealing with the microbes.
Okay, sir. Thank you, sir.
Thank you, Shesha. Thank you.
Thank you, sir.
Thank you.
Thank you very much. So let's move on to the next presentation by Dr. P. Maladi on recent trends in the management, in disease of sugarcane.
Yes, sir.
Yeah.
Yes, sir.
So recently I attended a meeting on the E.K. Janagamba Memorial. So I do remember about sugarcane reading station. And I met Professor Janagamma long back in 1970s when she was working at Madras Botany. And just a passing thought about Janagamma. That's all. Nothing to do with the pathology. But when I mentioned about sugarcane, I thought of Janagamma. Bye. Please, Dr.
1 minute, sir. So I will share, sir. Yes, sir. 1 minute.
Yeah, please.
Yes, sir. Thank you, sir, for your remembrance. Thank you. Sir, hello. Am I audible?
Yeah. Yes.
Yes, sir. Sir, respected chairperson, seniors, colleagues, and my dear student friends, good morning to all. First of all, I thank the organizer for having given me this opportunity to share my experience in this area. Actually, I am working for the past 20 years in sugarcane. Hence, I thought of sharing this brief. I want to brief this recent trends and management of fungal disease in sugarcane. 1 minute, sir. So, actually, as far as sugarcane is concerned, so far more than 250 diseases have been reported worldwide, and in India, it has been it was reported about 15 to 20 diseases, of which about 10 to 15% loss has been reported by all the disease on an average. But in endemic location, by threatening disease like red rot, we can observe even up to 100% loss we can observe. So in all the disease affects both recovery as well as tonnage of the crop. And these diseases are caused by different kinds of bioagents, causal agents, that is including fungal, bacterial, viral, and phytoplasmas. And irrespective of agents, invariably the planting material serves as the source of primary source of inoculum. And for the fungal diseases, the crop residue also serves as a source. Next one. Among the diseases, we are able to manage the viral disease in sugarcane by successful, that is meristem tip culture in combination with the virus induction. It is being followed under field conditions also in our laboratories, accredited lab, and regularly we are indexing the plantlets for production of virus-free settlings, and it is being regularly practiced under field condition in different factory areas. Hence, we are able to, at least for viral disease management, we could just say some solution for the management. Then regarding management of bacterial and phytoplasma disease, we are recommending the aerated stream therapy and hot water treatment under at a factory level, and for the manage and for producing the disease-free for distributing the seed material. That is at least disease-free seed material. For distribution of seed material, we are recommending this under field conditions under 3-tier nursery program. Then regarding fungal diseases, actually it is we are at different fungal diseases are there, among which the important ones are red rot, smut, wilt, and are the 3 important diseases. And to some extent, it's set out, and it is not it is a minor disease. And for this disease, we are tackling the 3 important diseases. We are tackling on different angles. The first, initially, especially for red rot and smut, we are mainly working on host resistance, and along with to sustain the variety, resistant variety or moderately susceptible variety, and to avoid the breakdown of resistant field conditions, we are working on conventional methods to sustain the varieties under field conditions, because whenever we release the varieties under field conditions, there is breakdown of resistance after some time due to the simultaneous development of frequent development of newer races. Hence, we are working on conventional approaches as well as molecular approaches to sustain the varieties under field conditions. Under conventional approaches, we are working on 2 aspects. That is, first one is identification of the effective molecules. It may be a fungicide or inducers or bioagents and the derivatives, or integration of bioagents and fungicides, and also the nano-based molecules. Then also we are working on suitable delivery system, because it is not a simple seed material for treatment. We cannot recommend the simple treatment methodology for sugarcane. Since it is a vegetatively propagated crop, it involves voluminous planting material. We have to go for some suitable delivery system for set treatment, as well as for secondary application. It is, if it is a short under young crop, for young crop we can recommend for spray but after some time we have to go for at least drip delivery to sustain the varieties. Under molecular approach also, we are working on identification and characterization of genes or proteins related to defense, pathogenicity, as well as antifungal nature of genes for genetic engineering approach, as well as to identify the potential target sites for better management. Now I will brief about some 2 slides. I will show about the molecular approach, what we are doing to identify the genes or proteins related to three aspects, that is pathogenicity or antifungal nature or defense related. Actually here we can see the upper part shows the two-way interaction and the lower part shows the tritrophic interaction with the trichoderma antagonist, and in which we are identifying and we have identified and characterized the volatile compounds as well as antifungal proteins and pathogenicity related proteins during two-way interaction, and as well as the genes expressed for cell wall degrading enzymes also we have identified, and during three-way interaction we identified defense gene expression, and likewise we are characterizing the genes. Here you can see the potential target sites like cytochrome P450 for azole fungicides we have characterized through this tritrophic interaction as well as by two-way interaction. Likewise, for the bacterial antagonist also, we are following the same technology for the same technique to characterize the genes by two-way as well as three-way interaction. Here we can see the by two-way interaction, we have identified antifungal genes as well as proteins as well as pathogens related proteins here. And this is about the small brief about the molecular approaches what we are following. And now further we are moving on based on this, we have identified the potential target sites for management with fungicides or inhibitors, and we are continuing this work. And also the identified antifungal genes and other defense genes, we are going for the genetic engineering approach. Then we will see about a conventional approach in detail, because molecular approach, many presentation we have done. So this is conventional approach, and this for under conventional approach, this is the this schematic diagram represents the integrated disease management for the fungal diseases. It starts from host resistance, physical method, cultural practices, biological, chemical control, and the combination of both, as well as induced resistance and non-obesity derivatives for the management of fungal diseases. And for as we all know, for the fungal disease, we have to avoid, we have to go for two aspects. That is, we have to see, we have to avoid the inoculum, otherwise we have to manage the inoculum. So to avoid the inoculum, that is primary source of inoculum, we are recommending for the disease resistant cultivars as well as disease free planting material. And for to manage the secondary inoculum, we are recommending set treatment as well as with different agents, that is physical, chemical and biological agents, and also along with the spray or soil rinse or drip delivery for the field application. Actually, earlier we used to recommend this soaking or swabbing with bioagents for the sugarcane seeds. Then if you see under field conditions, under farmers practice, even if you recommend, the farmers may not go for pre-treatment. Even if they go for treatment, they will go for only 30 minutes or less. But under our findings strongly supports says that the overnight soaking is necessary to for the effective infiltration of any molecules. Even it may be a fungicide or any other inhibitors or anything. So when we recommend the farmers, they could not adapt due to practical application of due to problem in practical application considering the voluminous planting material. So in that condition, we thought of having some mechanistic system for easy and effective delivery of molecules for the management of diseases under field conditions. So in that way, we developed a prototype having the based on the vacuum infiltration technique, and we found that by this technique, the delivery of fungicide was found to be equal as that of overnight soaking. So the mechanistic treatment as well as overnight soaking, that equally the delivery of material was found to be equal, and also the effect was also equal in inhibiting the pathogen as well as disease under field conditions. Since then we standardized this technique for vacuum level as well as duration and other things. Then based on this, we found that the fungicide, this method was found to be simple and also rapid, and it was found to be effective in delivering the material, and also it was found to reduce the quantity of material. That is, if you use 1 kg of material, that 1/10 of the fungicide was found to be reduced. By that, the cost was reduced, and also it was possible to deliver more than one agro input in this system. And by this was, and it was found to be compatible with other methods also. Hence, we found that we moved on to we filed a patent for this technology, and it has been published. And also now this year we have replied for the first exam. They have taken for first examination and the examination report, and we have replied for the first examination, submitted the reply for the first examination report. And then after that, we have gone for developing fabricating the bigger units for field application in collaboration with the CAE nearby, that is regional center here. And this is the first units we fabricated, and here we can see the difference between the conventional as well as mechanism, because in our institute for the management of the national hybridization garden, we are growing more than 600 genotypes, and to manage the wilt in this, every year previously we recommended only overnight soaking of sets in fungicide, but after developing this unit, then we compared this one, and we found that both were effective in protecting the sets from wilt, and there was no deleterious effect irrespective of the genotypes. Hence, recently, for the past 5 years, we are using this bigger unit for treating the sets every year, along with other fungicide and other nutrients and everything, for other agro inputs also for the management of wilt. And here we can see the fabrication of other units, subsequent units for field application, and we validated this unit under field conditions. This is in sugar factory areas for the management. We have conducted trials under field condition for the management of red rot and smut. And this slide shows the management of red rot using the mechanized treatment with thiophanate methyl and the bacterial antagonist Pseudomonas fluorescens. That time it was Pseudomonas fluorescens. Now it has been confirmed as Bacillus alvi. And this, and we could see the effective protection of sets from the soil-borne inoculum as well as improvement of yield. Then we also, then recently also we did under institute field condition, we found that the combination of, when we deliver the combination of thiophanate methyl and the Bacillus alvi in through set treatment device, it was found to be highly effective. Here you can see the improvement of plant growth as compared to other individual application and as well as inoculated control. So now we are recommending this for field application also. Then this is with respect to smut. Here we have taken the completely 100% infected smut material, and we found that by mechanized treatment with the propaganazole and along with the spray or drip delivery in the subsequent, then we could save the crop. Within 2 years, we could completely save the crop from the 100%. We reduced the infection from 100% to less than, that is the planting material was completely cured after 2 years. Then in 2-3 years we have continued, and then the same material has been used for the planting. Then after observing all the results, after seeing the results, effective efficacy under field condition, we thought of having we validated this unit for nursery application. And because nowadays we are recommending seedling planting instead of sets to avoid transportation, to avoid cost, to reduce the seed cost as well as to reduce the other seedborne pathogens also. Here, this settling planting, we validated the unit for treating single bud sets with different agro inputs. It includes fungicide and insecticide and nutrients for the production of healthy settlings. Here we can see this is the treated one and untreated one. Here the first quality settlings were found to be high by the treatment with this one fungicide, insecticide, and nutrients including urea, zinc, as well as iron. Then after studying this one with sugarcane, we could see that it is able to protect the crop under in nursery. It is highly suitable for nursery as well as main field application. Then we thought of validating for other crops. And just we validated for banana with the same input combination, and we found that there is a significant improvement in plant growth, and even and under field condition without any external application of other nutrients, anything. Now still after 1 year also, it was the growth improvement was highly maintained, and the bunch they're bearing the bunches in the treated one, but this untreated ones without any bunches, and since because we have not given any external application of any other nutrients, and it is very much shortened. And we also validated for cassava also. Here we can see the difference in treated and from the untreated ones. Then after studying all this, now we are mainly concentrating on the identification of effective antagonists, that is effective bioagent for developing the liquid formulation for effective delivery through set treatment device, because fungicides alone, we cannot recommend the fungicides for long time. So for to have bioagents alone or in combination with fungicides, we have to identify some effective fungicide bioagents. So in that case, we have identified effective bioagents like trichodoma and as well as Bacillus algae, and we developed the liquid formulation. And here this unit is highly suitable to validate any number of molecules and any number of in any number of planting materials, so that it has a provision to validate to screen the molecules or material in at a time. So we have screened different bioagents or also different derivatives and the metabolites, and we have selected the effective ones for field application. And here we can see the after infiltration, after treating the sets, we could see the growth of trichodoma in the treated sets as well as growth of Bacillus algae in treated sets in buds as well as in internal tissue. Then after confirming this one, further we move to synthesis of nanoparticles from the metabolites of bioagents, both from Bacillus as well as trichodoma. We are concentrating on the biogenesis of nanomaterial for field application, because we have the we have standardized the effective delivery system. So when if we have the smaller quantity of material, it will be highly effective for the large quantity of planting material. So we are concentrating on this now. Since we have proved this efficacy of this unit at a different level, and we have licensed this unit for a private entrepreneur, and now it is being marketed throughout India. Now, so far it is about 100 units have been installed in tropical as well as subtropical India, and they are effectively utilizing these units for main field application as well as for the production of disease-free settlings in healthy nursery under healthy nursery program. And also this unit can be fabricated for in different sizes for depending on our requirement. So these are some of the examples which we have developed, fabricated for our exhibition purpose or for other small for lab purpose and as well as greenhouse purpose and like this. So it is to have to avoid for easy handling. And then in continuation, we have upgraded this set treatment device for hot water treatment, and in which now we have standardized, we have fabricated the newer unit and a new unit, and we have validated this unit for treating the sets. Now, and actually this idea of this development is to have to for the varietal regeneration, because every year we have to regenerate our clones for under field conditions. For this we have validated this now under process. It is under process. We are validating this unit for field application as well as for regenerating the varieties to avoid non-fungal diseases also. So further it will be developed and it will be commercialized. Thank you.
Thank you, Dr. Malathi, for
Thank
your
you, sir.
work. Say here, I just a couple of doubts. What is the cost of the unit? What will be the cost of the unit approximately?
Including GST, it is about 65,000, sir. Including GST, otherwise 55,000. Sir.
55,000 for the treating how many sets?
Actually, single bird sets, it is 5,000 to 6,000 you can treat, sir. And for double butted sets means it will be around 1/10 it will be reduced. Actually, 500 to 600 only, because depending on the size of cutting. But a single bird sets, it is a single 1 acre for 1 acre. It actually 5,000 to 6,000 set by a single time. At a time you can treat, sir. And it is the time duration is it's 15 minutes only. But compared to overnight sex, it's only 15 minutes. We are giving treatment for only 15 minutes. I forgot to tell that it is a very rapid, only 15 minutes we require to treat the sets. So one entrepreneur nowadays, the entrepreneurs are using for this unit for 1 lakh seedlings they are developing every day. Or somebody they are using 10,000 to 1 lakh they are easily they are planting.
That's a commercially viable model. That's a very commercially viable model. 65,000 is nothing for a 15 minutes treatment. Then about the nanoparticles, what is the progress in the work of the nanoparticle use?
Now we got the project, DST project. We started the project and we are extracting the nanoparticles. In the subsequently, one oral presentation will be there. He will be presenting on the nanoparticle synthesis for biogenesis of nanoparticles for field applications.
Thank you, Madhu. Any questions from other participants? Hello,
sir. I am having one question.
Yeah, please.
Yeah, madam, you have. I am Dr. Angappan from TNAU. Madam, you have discussed about the metabolites from the biocontrol agents and their application for the management of set-borne diseases. I like to know that what is the period of protection offered by your metabolites, whether directly or in the form of formulation to the sets.
Actually, for metabolites, I have not formulated so far.
Okay.
And this bioagents we have treated, and it is up to 90 days. It is by artificial inoculation itself, we were able to protect the sets from by using this treatment with Bacillus salvi or trichodoma, or in Bacillus salvi in combination with the fungicide is highly effective. It is even more than that. It is it may not need secondary application if the inoculum is less. But for if the inoculum is more in the soil only, we are recommending for the secondary application. Otherwise, if the inoculum is less in the treatment itself, it is able to protect the plants.
Okay. Is it possible to multiply in some organic matters the biocontrol agent and apply? Then they can able to multiply in the soil itself to overcome the problem of the pathogen.
Whatever we are recommending, the farmers are not able to follow. That is the problem. So set treatment means only once if they do, then initial thing is it will take care of set borne as well as soil borne inoculum. So that is both the things. That's and it is easy for application also, and the cost effective. It is very cost effective. 1/10 of the chemical is reduced. Even the cost will be very much less. Within 1,000 rupees, they can treat everything. 1 acre or.
Okay. Thank you, madam. Thank you.
Thank you. Thank you.
Any other questions from others? Any other questions? If not, thank you, Dr. Malati. We'll move on to the next paper.
Thank you, sir.
That's by Dr. Veena from CTCRI on process and prospects of fungal disease management of tuber crops.
Malati,
yes, sir. Yes,
you have to stop sharing the slides.
Okay, stop. Stop. Stop sharing. Stop sharing. Okay.
Okay, thank you. Thank you.
Okay, thank you. Thank you, Veena. Sorry.
Dr. Veena.
1 second, sir. Respected Chairman, Co-Chairman, seniors and friends. Root and tuber crops are the third important food crop of man after cereals and legumes. These crops are very important and very unique because they constitute either staple or subsidiary food for about 1/5 of the world population. They have higher biological efficiency. Pardon? Pardon? They have higher biological efficiency, which range from 50 to 75 tons per hectare. They are climate resilient. That means they can withstand adverse weather conditions, and they have the capacity to yield in poor and marginal soils. Even though there are many tuber crops, I will be covering cassava, yams, taro, and elephant fruit yam, where fungal pathogens cause a serious threat to the production. Coming to the cassava, tuber rot caused by Phytophthora palmivora is a major problem in cassava growing areas of Tamil Nadu, which cause 50% yield loss. When in severely affected fields under conducive condition for Phytophthora, the crop loss can go up to 90%. In this case, no external symptoms will be visible on leaves and stem of infected plant. So the farmer will not be able to identify the diseased plant. Dark colored round to irregular water-soaked lesions develops on the mature leaves. The lesions enlarge, causing internal browning, oozing of internal fluids, and finally the shriveling of the tubers. So the farmer will come to know only at the time of harvest. When he lifts the plant, there will be nothing and it comes off very easily. Infected tumors emit a foul smell and rot within 5 to 7 days. And coming to the management strategy, avoiding excessive irrigation and improving drainage condition, which helps a lot to reduce the population of Phytophthora. Water stagnation, which is conducive for this pathogen, can be avoided by breaking the hard pan. This is the way, the hard pan. By using chisel plow to make 15-inch deep plowing will enhance the water percolation and thereby reduce the pathogenicity. Other management strategies are we have to follow strict field sanitation because the pathogen surveys in the infected tubers under the soil. So we have to, it's very important to remove the affected tubers. And planting in ridges, crop rotation, addition of enough organic matter and trichoderma. This is our management strategy, and it has been found that this strategy is, because of this strategy, the disease is under control now. And there are some emerging problems. This is tuber cracking. This is the very recent one. Maybe last 2-3 months only we started seeing it. And because the farmers, they have sent many photos to us, and we have also some of the field we visited and saw this. So however, we could not find any pathogen associated with this one. So in our institute itself, the plant and soil nutrient analysis and physiological factors, so all are looking up now. So I think we will be coming to know about what is exactly is causing the problem. Another important problem which is becoming very serious is root rot. So this actually it occurs in 3 stages. Before rooting itself, the decaying of sets along at the collar region it starts. At 2 to 3 months old plants, yellowing of the leaves and drooping of the outer leaves, and stem and tuber rotting, you can see it. When the crop reaches almost 6 months, stems decay at collar region and rotting of the tubers. According to the farmers and our own view, what we are seeing that it is only seen in the field where it is the paddy field is converted for cassava. And coming to the etiology, we suspect involvement of 2 or more pathogen, or maybe it's like a nematode or something else will be. So this is actually, we are giving high priority, and this project actually it is looking after by Dr. Jeeva. And initial finding confirms that Fusarium species is the one of the causative organism. So we have made an ad hoc recommendation, including strict field sanitation, crop rotation, and provision of good drainage, addition of lime, neem cake, and another one. It's actually it is all the tuber crops. It's all it's clonally propagated. So set treatment, if the pathogen is affected the sets, it will go to the next crop also. So set treatment is very important. So we have developed a set treatment with the carbendazim or combination fungicides, carbendazim and mancozeb. Excuse me, some problem. Just give me a minute. I will just rectify it. Hello? Sir, am I audible, sir?
Yeah,
Yes,
yeah.
clear.
Sir, is actually the slide is not moving. Just give me a minute, I will rectify it.
Okay, okay.
What to do? Sir, kindly go with the oral presentation. I will just rectify and let you know, sir. Sir, kindly go with the oral presentation. I will just rectify and call you back, sir.
Okay, Bigran. So, rapporteur, can we give me the list of oral presenters so that if I can start with the first presenter? Hello.
Sir, first presenter is Jayalakshmi. Hello. Sir, list it is in mail.
So I think we can. Is she ready? Jayalakshmi, are you ready? Are you ready with the presentation? Okay.
Mail Madhunde അയച്ചിട്ട് അല്ല എനിക്ക് ലോഗിൻ ചെയ്യട്ടെ
Yeah, I think we'll start with the Jayakshmi's presentation. The evaluation of eco-friendly strategies for the management of grain discoloration in rice. Say, Dr. Jayakshmi, are you ready? Dr. Jayalakshmi, can you hear me? Professor Anagapan, can you hear me?
Yeah, yes, sir.
Hello.
Sir, you are
So
audible.
why can't you call?
Yeah, you are audible.
Then why can't you call Jayalakshmi? I wanted to call her for the presentation.
Okay.
So I think she is not hearing me.
Dr. Jayalakshmi? Dr. Jayalakshmi?
No, I think Veena has to.
Dr. Jayalakshmi?
I think
her audio is not connected, it seems.
then call the next person. We don't waste time.
Sir, next presentation is by Dr. Tirubonamala. The topic is innovative approaches for low-cost cultivation of paddy straw mushroom for coastal area and different cropping systems.
We can call her.
Sir, I called her, but she did not pick up the phone, sir.
So, what about the?
Sir.
Did you call Dr. Jayalakshmi? Please call Dr. Jayalakshmi
Yes,
over the
sir.
phone.
I called her.
It's open. The Jayalakshmi's presentation is open here.
Sir, again I call you, sir. PPT open பண்ணுங்க. இருங்க பண்ணுங்க. இருங்க.
Dr. Lata? Dr. Lata?
Sir, I call just 1 minute, sir.
Yeah?
Checking the பண்றேன் sir, checking the PowerPoint.
Okay.
No, PowerPoint is she's on the screen.
Yes, sir.
Her PowerPoint is.
She has to present. Her PowerPoint is shared already.
Yes, sir.
She has to present.
1 minute waiting.
Is her audio on?
Yes, sir. Audio on, sir, but
Dr. Jayalakshmi?
I think we'll go for the third person now.
No,
Sir,
but we could not able to.
Screen is closed. Next cannot do, sir. That is the problem.
The sharing could not be done.
Yes,
sir.
sir. Yes, sir.
She has to close.
She has to respond.
Can admin do something about it?
Yeah.
Dr. Jayalakshmi?
Her audio and video are both on.
Yes, sir.
the madam is cut. Next person.
It was disconnected by admin. Admin has disconnected it. Now you can continue that.
Then we going for
Dr. Tirubonamala?
Yes, sir.
Yeah, you can start.
Yeah, I'll just share, sir.
You can present.
Yes, sir.
PPT file எதுல இருக்குங்க?
இல்ல அந்த ஓபன்ல இருக்கு பாருங்க.
IPS presentation file.
இல்ல இப்ப மேடம் ஓபன் பண்ணிருந்தாங்களே. எங்க மேடம் ஓபன் பண்ணாங்க?
ஆமா, ஃபைல் ஓபன் பண்ணுங்க.
ஃபர்ஸ்ட் ஃபைல் ஓபன் பண்ணிட்டு அப்புறம் வந்து
now You see, we
Yes,
don't,
sir.
we have got only, now it's almost 11:15, 11:10, 11:15.
Minimize
So
பண்ணிட்டேன்
we have 10 papers.
Final. Click பண்ணுங்க.
இல்ல தம்பி இல்ல அது மேக்ஸிமைஸ் பண்ணிருந்தேன்.
Share ஆயிடுச்சு.
Share the.
Back click பண்ணுங்க. Share ஆயிடுச்சு.
Sir, is it visible, sir? Sir, are my slides
Yes,
visible?
visible,
Yeah.
visible, ma.
Yeah,
Not
okay,
the
sir.
slide, only the folder is visible.
Sir.
You can click on the
Only
PowerPoint.
folder only visible, ma'am.
Hey, Sangeeta.
Not PPT. Yeah.
Only folder is visible.
Please click on the PowerPoint.
ஆடியோ வரல.
Madam, you open your PowerPoint.
Sir, I opened, sir.
No, you have not.
You have not shared then.
I have shared. Then.
Madam, after opening your PowerPoint, then share it.
Please copy paste that PowerPoint in your desktop and then open it.
Zoom நினைக்கிறேன்.
இதுங்களா மேடம்? இதா அப்ப
Sir, now it's visible?
No, it's not visible.
Just a minute, sir.
Opening. Oh yeah.
It's okay.
Yeah. Now it is visible.
Now
Work.
that's okay.
Sir, already I've shared, sir. Can you see, sir?
Yeah, it is.
Now
not
we
there.
can see.
Okay,
Now we
okay.
can see.
You can start from first slide.
Yes, sir.
You can start.
From
Yes,
first slide.
please.
First slide.
From first slide. Please, you start from first slide.
Back click. Close பண்ணுங்க.
Please go to the first slide.
Sir, now it's visible, sir?
No.
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 Ma'am, only folder only visible, ma'am. PowerPoint not open, ma'am.
Madam, open your PowerPoint, make it in a slideshow mode, then you
Yes,
share
sir.
it.
இதை க்ளோஸ் பண்ணுங்க.
I think you have to close it. Just a minute, sir. I'll close all the other. Close it. So now, sir.
Now go. Yes.
Okay. Okay, sir.
Yeah. Now it's.
Thank you. Okay.
Go to the first slide.
Oh, yeah. First slide,
Go to
sir.
the first slide. First slide.
This is first slide, sir.
Yeah. Yes. I'll start with that.
Yeah.
Please.
So, good morning, respected chairman, co-chairman, senior friends, colleagues, and my friends, my dear friends. So, good morning to one and all. Myself, I am Dr. G. Tirubonamala, Associate Professor, Department of Plant Pathology, Tamil Nadu Agricultural University, Coimbatore. So, I am going to present my research paper on innovative approaches for low-cost cultivation of paddy straw mushroom, Volvarilla volvaceae, for in coastal areas and under different cropping systems. So, the Volvarilla volvaceae paddy straw mushroom is the third most important mushroom in the world and good flavor.
நீங்க யார் பேசுறீங்க?
Good flavor.
Okay. நானே அவருடைய சிஸ்டத்துக்கு ஒரு மெசேஜ் மட்டும் அனுப்புறேன். அவர் வந்தா கொஞ்சம்
Owing to its good flavor and taste. So, however, this mushroom is its commercial cultivation is infant in the world. Maybe China is the leading producer, but in India, in the year 1940, Su and Seth, they have worked in 1923, I think they have worked for some time on these mushrooms, and later in 1943, Thomas and his co-workers, they were at this Tamil Nadu Agriculture University, previously Agricultural College. They have worked over here and they have just attempted some work on the cultivation of these mushrooms. And later,
our previous Vice Chancellor, Dr. K. Ramasamy and Dr. Rangasamy, and both of them, they have also attempted to some work on this Valderilla cultivation. But then our student, one of our student, Dr. Sangeeta, has attempted the cultivation of these mushrooms in indoor by developing this circular compact bed method. But however, this indoor cultivation is not cost effective, as the bioefficiency of this mushroom is very less. So if we see, though the climatic conditions prevailing in India is quite suitable for this cultivation of this mushroom, because it is a tropical mushroom and it requires a temperature of 30 to 35 degrees centigrade with a relative humidity of 80%. And it's one of the tastiest mushroom. As I said, that this is the only mushroom that comes to harvest within 10 days with a shorter cropping cycle, maybe at the maximum of 17 to 20 days. But however, if we see, it's having a very low bioefficiency of a maximum 20 to 23% only, because of the reduced secretion of lignolytic enzymes by this mushroom. It is not able to colonize the substrate fully and to utilize that substrate. So due to owing to its low bioefficiency, its cultivation in indoors, TNA has developed a technology in 2002 itself for cultivation of this mushroom in indoors using polyhouse, like how we cultivate milky mushroom. And then we have developed a circular compact bed method, and we can see that maximum bioefficiency is only about around 17 to 19% or at the maximum 20%. So in India, if we see, only certain parts of Chhattisgarh and Bhubaneswar, they have started this outdoor cultivation of this mushroom, because the microclimate there is quite suitable, and also the paddy straw, this mushroom will grow is only on paddy straw. It prefers only paddy straw, and even because of weak colonization of the substrate, we cannot use any other substrate to tap maximum bioefficiency. So in India, if we see, there are no standard cultivation techniques like for other mushrooms that you have to cultivate this mushroom only by this method. So if we see, that is the need for the study, for which I have taken this study to develop some approaches so that we can take this mushroom to the commercial level, so that it will be, the farmers will gain benefit out of this, so that the paddy growers in the delta and coastal areas also will be utilizing the paddy straw for cultivation of this mushroom, so that their resources can be effectively used. As well, they will be getting additional benefit. That was the motive of this study. So earlier, myself and the present registrar and Professor Dr. Krishnamurthy, we have attempted to some works with our previous director, Dr. Marimuthu sir, who has motivated us behind this work, and where we took up this mushroom for cultivating this mushroom as intercrop in maize, because at TNAU, we attempted a work previously in maize hybrids, which has a cropping cycle of 105 days. So at the 45 days silky stage, the same microclimate of this 35 to 35 degrees centigrade with a humidity of about 75 to 80% will be attained in that microclimate. And we prepared some mushroom beds with the bundle method, and we placed, and we could see that from 45 days until 75 days that microclimate was available, and we could able to harvest the mushrooms. So we thought of carrying over with this prelude idea. We wanted to take up this mushrooms to the commercial level. So having experience with this or success with this project was formulated with an objective to find its suitability of paddy straw mushroom cultivation in maize, comparing it with banana and coconut cropping systems, where such climate is again, microclimate is possible, and also to test the outdoor cultivation of this paddy straw mushroom in rice growing coastal and delta areas, so that effectively the resources can be used with low cost. So the methodology what we adopted, there are a lot of methodologies for paddy straw cultivation. So what we slightly tuned this is, without steaming or autoclaving the paddy straw, it can be soaked in lime water solution, 2% lime, for 8 hours, and then the paddy straw can be taken, and then open bundle method, the bundles can be prepared using 4 kg paddy straw per bed, and then we are using the paddy straw spawn is prepared in paddy straw itself, so that easy colonization will be there from the paddy straw spawn to the paddy straw itself. There are a lot in Bhubaneswar and Chhattisgarh, they are using wheat and also rice based paddy spawn. Even we tried with many other spawn, but we found that this paddy straw spawn is quite suitable because it easily colonizes. At the rate of 2%, we are using that spawn and amending with a little horse gram powder, and then the beds are covered with thin polythene sheets for 5 days. And then observations are the days for spawn running, days for pinhead formation, days for first harvest, the cropping cycle, as well as the the daily bed temperature and the microclimatic conditions are also recorded. So if we see, So just proceeding to the results of the study, what we have done, we used this potential strain of Varroa valvaceae. We have many strains with us, and we selected this VV4 strain from the selection from our ICRIP project. Actually, this strain is giving bioefficiency of 18.6% to 20.5% throughout the year. So if we see that identification of potential strain is more important, because not only the yield is, we cannot take it here because this mushroom is very poor and unstable yield is there. Apart from that, it's so delicate that once it comes to harvest, within 6 hours, the mushrooms are getting deteriorated and the quality goes down. So, but this strain, this is the different stages of the mushrooms. You can see this is the, after the pinhead stage, after spawn running, you can see the egg stage of the mushrooms. Then the morphogenesis comes like an umbrella, they just open, and after that the mushrooms are fully bloomed like this. So we can harvest these mushrooms at this egg stage, or we can harvest at this umbrella stage also. But you can see that from the egg to harvest stage, this strain gives 1.8 days in the bed itself, and then you can see the retention of the open mushrooms on the bed also. It retains for another 1.6 days, which is a boon to get this strain. So that under storage conditions, normally we cannot store Varroa under storage under refrigerator, but this mushroom, it can be stored under egg stage in airtight containers in refrigerator for another 1.51 days. So we have taken this strain for the commercial, for these trials. So if we see what I have done is, I have tried this Varroa valvaceae mushroom cultivation in banana cropping systems from where the crop was at 4th to 5th month stage, and we prepared the beds as I have mentioned in the methodology and placed the beds in the interspaces, and we can see that the average bed temperature with a microclimate temperature of 39 degrees centigrade and the relative humidity of 85.5 was recorded during the cropping period of that 16 to 17 days. The spawn days for spawn running was completed on 8th day, and pinheads appeared on 9th day, and the days for first harvest also began on 10th day with a bioefficiency of 20.6%. Similarly, and I have conducted this trial in maize also cropping systems. So we can see that at TNAU the trial was conducted in this August to September. Actually, this project was taken as a short-term project through the project the action plan was proposed, and our Vice Chancellor was so keen that some technology should go to the delta farmers, and as well as for conversion of the natural resources like paddy straw into the mushrooms, and to get the additional benefit for the farmers. So with the motivation from our Director CPPS and our present registrar and my professor Dr. Krishnamurthy sir, we formulated and took up this project in the short period of this 2 to 3 months, and came out with these findings that in the maize cropping system also, we can see that the average bed temperature with a microclimate of 37.5 degree and relative humidity of 82.5% was recorded throughout the cropping period, with a spawn running period of 9th day and 10th day, and the mushrooms came to harvest on 11th day, and the entire cropping cycle was completed by 17 days with a bioefficiency of 19.4%. Similarly, in coconut cropping system, we tried at our TNAU, but we could see that though the bioefficiency was attained to 19%, there was further delay in the first harvest by 1 or 2 days because of the relative humidity was about only 77% was maintained, not above 80. So that there was further delay because of the open, you can say the humidity is not there because of the interspaces is more. If we are going with other short hybrids with the closer spacing, still we could get more yield. So this is another finding. So after having successful cultivation of these in banana, coconut and maize, we found out that banana is quite suitable when compared to all, followed by maize and then coconut. So recently, in this November month, I have conducted a trial in the banana coconut croppings intercropping system at Vedapatti in Balagondar a farmer's field, and where I could see that in this banana intercropping system also, here the average bed temperature of 37 degrees centigrade, the relative humidity of 83.5, similar to the indoor cultivation, was achieved with that in that cropping system, where the mushrooms came to harvest on 12th day with a total cropping cycle of 17 days. And here you can see the children were also at the corona period, they were at home and they were also involved, and they were quite happy to see the mushrooms, and they all enjoyed, and they are now asking for the spawn to take up this project, this study. So having tried with this, now what I did after completing the TNAU, then I went to the coastal areas at Pumbuhar. I was there on 6th there, and I have given training. I invited farmers from Tiruvarur, Keralam, Pumbuhar, and from Kumbakonam and Velugudi, and I have given demonstration to the farmers on this importance of this mushroom. They are aware of this mushroom because it's a locally available mushroom which is there in the nature, but the only thing, the commercial technology has not reached them, or we have not promoted, we can say, because of the reasons which I said prior. So I gave them the technology, and I taught them how to do, and I had took my one of our permanent unskilled Mazdoor with me, Ms. Shanta, who was also showing keen interest with conducting the demonstrations, and I provided the spawn to them. And again, I visited the second week. So I went there from October to November, 3 times I went over there. And then again, I conducted the trial there itself in the farmers field in banana ecosystem. And we could see that the same 7.5 degrees centigrade of temperature, relative humidity of 83.5 was achieved with a good cropping cycle of 17 days and with a bioefficiency of 18 days. And he's a proprietor of Priyam Mushroom Farm. And so he has sold this mushroom as a elite mushrooms for about 200 rupees per kg. And he could get a cost benefit ratio of 1:2.8, which is really remarkable, because in oyster also we get only 1:3.2, not more than that. And then we can see within a short cropping cycle it's possible. Then I conducted a trial in Mr. Rajendran at Poombuhar, which is close by Karaikal, this area. And then, here also the success is achieved. We can see within a cropping cycle of 14 days, since the climate is very close to the nearby seashore, within another 5 kilometers is the seashore. So the climate was so conducive, the microclimate was there. So within 5 days, the spawn running was completed. And within 6 days, we could see the pinheads, and the mushrooms were harvested on 8th day with a good bioefficiency of 19.2%. And also he also got a bioefficiency of a cost benefit ratio of 2.9. And another farmer at Kumbakonam, which is a delta area, rice growing areas. And here I conducted a trial, and there also we could see that the bioefficiency of 18.6% was achieved within 18 days cropping period, as in other areas with a good microclimate also was achieved. He is actually having a shop nearby his farm itself, and where you could see that that day I was there by 7 o'clock in his field. We can see a lot of people there. They came, and his daughter and son, who has completed BCom, is now they have started this business in Corona, and they have 40,000 out of this. And so you can see that because he has now converted mushrooms for his value addition as elite mushroom product, and he has developed a good mushroom value added product out of this, and he has earned a bioefficiency of 1:2.8 out of this. So we can see overall, I can say that whatever be the cropping system, or whether it is cultivated outdoor in the coastal ecosystem, we can see that the environmental conditions are like this. But here we can see the bed microclimate is again 5%. The temperature is increased, which is more conducive like the indoor cultivation temperature. So that like our indoor cultivation itself, we are able to achieve the bioefficiency of around 18 to 20.6 like the indoor cultivation, but here no cost is involved. So the farmers are able to convert their the natural available resources without less cost into the mushrooms. So I can say that conclusion, I can say that there is scope for commercial cultivation of this paddy straw mushroom in Tamil Nadu, and it is possible to introduce one more mushroom for edible mushroom for the farmers and the market for the consumers. And this best fits in the integrated farming system. And also there after the mushrooms are cultivated, the spent mushroom substrate can be converted into biomanure. Thereby we can ameliorate the soil by applying this biomanure. And so my future studies are directed towards popularization of the outdoor cultivation of this technology of this paddy straw mushroom to all the entire area of Tamil Nadu, so that we can take up this technology to the next level. So I sincerely thank the university for providing me the facilities to carry out this study through my university project on improved techniques for commercial cultivation of paddy straw mushroom. I thank again to the farmers also and the team members, Dr. K. Angappan and my JRF Arun Prakash, and for carrying out helping me in supporting me in this study. So once again, I thank the organizers for providing me this opportunity. Thank you, one and all.
Thank you, Thirumalai. Say yes, with the 2.8, it's a fairly good proposition. But one question: do you notice any insect damage? Because if it is outside.
No, sir, not at all. Only ants were there, sir. Why these black ants? They came because we used to amend with 2% horse gram, sir, for as an amendment, because to enhance the mycelial colonization. That time I could find only in 1 or 2 beds few ants were there, black ants, but they did not disturb the mushroom growth.
If it is so, but it will be an ideal thing for rubber plantation, because with 38 degrees Celsius, it's ideal temperature inside rubber plantation during the summer will be ideal, and RH also is almost the same tune. So
yes, sir.
Be additional income for the rubber growers.
Certainly, sir. I wanted to take this project to this study to other crops also, cropping systems also. I would like to study, sir. As you said, I'll surely try. Yes, sir. I will try, sir. Surely.
Thank you. Thank you.
Thank you,
So,
sir.
I think we'll take up the paper by Dr. Veena. Her paper is ready. She has solved the problem. So we'll move on to Veena's paper. Dr. Veena?
Okay,
Thank you,
I'll
sir.
just, I will close it, sir.
You please close it. Thank you, sir.
Thank you, sir. Thank you, members. Thank you.
Thank you all for the patience. Actually, I was talking about the emerging disease on cassava. Actually, this particular disease was started noticing from after 2018 flood. So the disease, I think up to this point, I have already explained. So we have developed one management strategy also, and we will go to yam. The most important disease in yam is the yam anthracnose caused by Coltrutrium gliosporoids in Dioscorea alerta, that is in greater yam, which is highly susceptible.
Your PPT.
Madam, your PPT is not visible.
Please share your PPT.
You
Dr.
forgot
Veena,
to share
please
it.
share your, yeah.
Yeah. Now it's ready.
Okay. And thank you. So we will go to the yam. The anthracnose caused by Coltrutrium gliosporoids is the most destructive disease in yam. Dioscorea alerta, that is greater yam, is highly susceptible, which cause almost 30 to 60% yield loss in yam. So these symptoms differ with the cultivar. The disease starts as small, dark brown or black spots or lesion on leaves, petiole, and it also affects the petioles and vines also. Often this lesion is surrounded by a yellow halo. Otherwise, sometimes it will be restricted. Otherwise, it enlarges and coalesces, resulting in extensive necrosis of the leaves, leaf fall, and dieback of the vines. The withered leaves and stem dieback give the plant a scorched appearance. So the disease is also known as scorched disease. So in coming to the management strategy, in all the management strategies, we give maximum emphasis for developing resistant variety. So for the host resistance, all our germplasms are being screened for the susceptibility for this yam anthracnose. This is the in vitro technique we use for the screening. Then the second stage is, once if it is some result is coming, we will go for the next stage in small pots. And after that, it is field screening. These are Sri Kirti, Sri Kartika, and Orissa Elite. All these three are released from our institute, that is CTCRI. Here you can see Orissa Elite is highly susceptible, whereas Sri Kirti is tolerant, and the Sri Kartika, it is not at all taking up the infection. And coming to the biological control, apart from the trichoderma and all isolates we are doing it. We have isolated some endophytes from the 10 medicinally important plants, and we got many isolates from there. Based on their performance, we have selected 7 bacterial endophyte and 3 fungal items. They were molecularly characterized. Once again, we have done selection. Finally, we went to the Bacillus licheniformis and Bacillus amyloliquefaciens. This is the pot culture experiment with this licheniformis and Bacillus amyloliquefaciens, and this is the result. This is the among the treatment, Bacillus licheniformis tuber treatment and spray, that was the one which was giving the least disease severity. So now the field studies are in with this treatment. That research is going on, but we do have a package for the farmers. Here we give soil and tuber treatment with the trichoderma coupled with the foliar spraying of carbendazim, that is 0.05%, 7 times we have to give. So this here, the initiation of initiation of a symptom 3 times at 15 days interval. After that monthly interval, we have to give the spraying. And coming to the taro. So in taro leaf blight caused Phytophthora colocaceae is the most destructive disease in taro, which is this, we do consume leaf
petiole, and the tubers. So this pathogen is capable of infecting leaf. There it causes leaf blight. In petiole also, this actually it will break. Once it is severely infected, it will break. And tuber also, it causes in field and in storage also, it causes. And it can cause corm yield up to 50% and leaf yield by 90%. That much destructive is the pathogen. So this is the taro leaf blight symptom. This just start as small light brown spots appear on the surface. Then the spores enlarge rapidly, becoming circular, sonate, and purplish brown to brown in color. Spores increase in size, coalesce, and quickly destroy the leaf. In some cases, here you can see that bright orange or brown plant exudate ooze comes out from the infection site. Actually, what happens with the taro leaf blight, if the normal leaf, it lives for 30 to 40 days, here the lifespan will be reduced to less than 20 days. In our epidemiological study, what we found, if the rain or if the dew is there, the leaf, means once it's get infected, within a week, the complete destruction occurs. Otherwise, if these two factors is not there, even if it is getting infected also, the spores will be restricted. And functional leaves also comes down from the normal, it is 5-7 leaves are there. Here it is come to 2 to 3 leaves. And here in this case, in the host resins, many workers has developed many varieties. And now from our institute also, 2 varieties we have released, that is jangri, one just renamed as Bukripa and Muktakeshi. And many workers from many centers, they have these 2 are giving very good, with showing very good resistant reaction to TLB. And the last 7 years, almost 400 accessions, whatever the germplasm we had in taro, is screened under field conditions. So when in field, whichever the accession showed some resistance, once again we have challenge inoculated by growing it in pots. These 10 accessions, these are the 10 accessions, they are continued to show the resistant reaction for the last 5 to 6 years. Now our crop improvement division, they are just evaluating for the other agronomic practices. Once that agronomic traits, so that is also good, this will be released soon. And with an idea to minimizing inoculum, we do advocate the removal and destruction of infected leaves and tubers, because this pathogen, it survives on the leaves and the tubers. This is actually the edible part, and for the propagation also we do use the same thing. So once it is infected in the field, we have to take care, and we should not leave any tubers in the field, so the infection will be more. And use of disease-free planting material, and there is a practice of using mother combs also, some people. So this should be wherever the disease incidence is there, they should avoid planting by mother combs. And shifting a planting date or delaying planting, because this TLB is very severe in the rainy season. So if we can shift or either advance or delay the planting, maybe the critical period we can avoid. And intercropping and short duration crop, this is Stelia. This is actually very short duration crop. It is only 3 to 4 months it comes. So this is, suppose if we are planting this one, we can adjust, we can avoid the high rainy season. And what we have seen, initiation of the disease, how this initiates in every season. So what when if the previous crop, suppose if it is the previous crop is infected, so the lowermost leaves, or that is the leaf touching the ground, it gets first infected, and from there the disease advances to top like a ladder. So with the mulching, either with the organic material or the black mulch, whatever it is, so mulching can delay. It's not absolutely we will get the infection, but it can delay the incidence. Coming to the biological control, these are already reported. Apart from this, we got many Bacillus isolates, and these are all where just we have done the pot culture studies also. There also this Bacillus amyloliquefaciens, subtilis, and licheniformis. These are all increasing the yield. This is pot culture study only, and they are reducing the PDA also. And there is one particular problem is there, because this TLB, this occurs during heavy rainy season, and this taro leaf has a waxy leaf coating also. It makes the fungicidal products ineffective. And one more thing, there is most of the places that this taro is grown in paddy fields or wetlands, so that spraying or any application at the rainy season will be difficult. So we thought, why can't we see whether just giving at the scotch treatment and the soil treatment alone can we control or can we manage the disease? This is the result. Even just giving the basal treatment alone, So to a certain extent, we can reduce the incidence. If we cannot take any spraying or other operations during rainy season also, we can do it. Piriforma spora indica. This is another root endophyte. This isolate we received from Amity University. This also we have tested. Definitely it is well known that it is a growth promotion is there. And for the first time, we confirmed its colonization in taro leaves by confocal imaging, P8 gene amplification, and trypan blue staining. And we are seeing the disease suppression also. In both, we used both Srikiran, that is susceptible for 5-TLB, and Muktagishis, as I told, it is resistant. In both cases, it can reduce the infection. And what happens after the colonization of taro leaves? So we have challenge inoculated with Phytophthora colocaceae, this already P indica colonized leaves, and we have seen that increase in chitinase, glucanase activities and total phenol control, and the differential expression of genes consequent to P indica colonization was also studied, and 5 genes were identified, and the functions also were annotated. So 4 of them, this comes under defense related pathways. Then, as you all know, that actually tuber crops we are giving very much important, because actually we want to go for the 100% organic. That is slowly we are shifting. So another method also, we have some organic amendments. We are using so much organic matter in the tuber crop production also. So here in this case we have used vermicompost. So here the cormal treatment we gave with vermi wash, and soil application with vermicompost we get given, and then dredging and spraying at 60 days and 90 days after planting with the vermi wash. In this case also, we can reduce the disease to a good extent. We can reduce the disease incidence. Coming to the chemicals, these many fungicides, many people are reported about copper fungicides, metalaxyl, borax, these all were reported. We have also compared this one. Here also again, this metalaxyl, that is mefenazole, it is giving very good control. That is the best one. We are also found that even potassium phosphate and our vermic wash treatment also can offer very good treatment. This is our present management strategy: grow the resistant variety Muktagashi, strict field sanitation, mulching with paddy straw or locally available organic matter, and treating the comers before planting with this point is very important, and most of the farmers or most of the recommendation we don't see. Actually, the planting material itself we have to treat it, because it's from the crop to crop it goes. This point is very, very, very important. And you give a prophylactic spraying with any of the fungicide and repeat the application in 15 days interval only during the rainy season. And suppose some farmer, they want an organic management strategy, that also we have developed. Here, instead of fungicide, treat the cormals in cow dung slurry mixed with trichoderma, 5 gram per kg of cormal before 2 days of planting, and just stray dry. And after that, soil application of vermicompost. This point I told that application of vermicompost and vermicompost, and then you repeat application of the same at 15 days during rainy months. This is another emerging problem. Actually, this is not taro. It is a related crop. This is Sandosoma. Here, we never had any problem, only except the deficiency disease, we never had any problem. But this particular season, we got many queries from the farmer. Actually, we could not inspect the field because of the COVID restrictions or not. These are the pictures sent by the farmers, and about we are suspecting Pythium involvement. So we have recommended this field sanitation with proper drainage, methyl mango drenching with 0.1% and copper oxide. And we got the feedback, it really controlled the disease. And we'll go to the last crop, that is elephant foot yam. This collar rot caused by Sclerotium rhodesi is the most destructive and common disease in all elephant foot yam growing areas. So usually this prefers rainy season followed by warm weather. And usually the later part of the crop is very much susceptible. However, it is capable of attaining any stage it can come. And injury caused at the collar region during intercultural operation is the predisposing factor for the disease. This is the signs and symptoms. So it starts at the collar region as water-soaked lesions, because it's the translocation of the water and other things restricted. So yellowing of the leaves it forms, and finally the plant collapses. Here you can see the clear growth of Sclerotium rhodesi at the collar region. In elephant footium, so far no assertions have been found with any resistance. This we won't be able to, because if any field screening will not give it, unless and until it is sick field, we cannot take. If one plant is just surviving also, we cannot take it as a resistant in case of sclerotium rosei. So we do like challenge inoculation, we do. This is the material. This is elephant footium itself. We will multiply and we will change inoculated. In the, after all this, only 3 exceptions. This is wild exception: AM1, AM128, and AMW22. These are the only 3 exceptions show some tolerance to Sclerotium rosei. Here also, so many isolates of bacteria and trichoderma. These are almost 43 very good trichoderma we have. We have compared all with, and just we have taken this beta glucanase and chitinase induction and all the antagonism as a criteria. And finally, with all the compiled one, these 5 isolates, these are we are using for the multiplication and recommending for the our control measures. Here also, when you talk about the organic emulsions, the same way, the seed treatment, that is not actually here, it is cormal treatment plus soil application and drenching at 90 and 120 days is the desired result. And finally, with all this, this is the recommendation we give, that is use disease-free planting material. Treat the combs in mangosub carbendazim, that is combination fungicide at the rate of 2 grams per kg before planting, removal of infected plants and providing improved drainage, drenching the plants twice with the same fungicide we have to give, and remove the infected plant, give additional drenching to surrounding plants with the combination fungicide. So another separate strategy we have for the organic growers. Here in this case, the treat the combs 3 days before planting in cow dung slurry amended with the Trichoderma aspergillum. Application of Trichoderma aspergillum enriched farmyard manure or vermicompost, whatever it is, you can do it and give apply at the time of planting. And once again, application of Trichoderma aspergillum incorporated vermicompost at the collar region immediately after intercultural operation. This point is also very valid because, as I told, that it's an injury caused at the collar region is very important, and most of the time the injury is caused during the intercultural operation. So don't wait for any time. Immediately after intercultural operation, the plants has to be protected. And removal of infected plants and additional application to surrounding plants with the Trichoderma aspergillum incorporated vermicompost. So this, I was talking about the field diseases, and we have another serious problem, that is post-harvest rot in elephant footium. Just see the fate of the comb. So we identify 15 pathogens found to cause post-harvest rot in elephant footium. These are the pathogens. All the pathogens were characterized and identified, and we could see 4 new genera, that is Cunninghamella, Ceratobacterium, Coltutricum, Gliosporid, and Penicillium. This is the first time we were reporting from the elephant footium. Similarly, 3 more species, Rhizopus oryzae, Fusarium brachygibosum, and Fusarium oxyspora. These many pathogens also cause the post-harvest rot in elephant footium. So we have the strategy, management strategy also we have. This is the organ like Trichoderma. Then chemicals is there. This is the biopesticide, that is the biopesticide developed from cassava leaves from our institute, and the combination of all these things. So based on that, if this recommendation we have, minimize injury to combs during harvesting, transporting, and storage. Carefully remove infected portion from the comb. Always store in a ventilated place. Dip the combs in this combination fungicide we are given. For the organic growers, actually, as I told Ariel, they can mix with the trichoderma. They can dip the combs in cow dung slurry amended with the trichoderma, and then that. This has to be done just immediately after harvest, and otherwise you will be losing so much of your produce. After treatment, dry the combs under shade and store. In elephant footium also, another problem, it is coming as an emerging problem. Some you can, I think most of the people, and there is a report of phytoplasma also in elephant, it has come from Dr. Ravu's lab. So there, but we do get with what is rotting of the leaf as well as the pseudostem without yellowing also. So we have made a survey also, different kinds of symptoms we could see it, and we could prove the pathogenicity of Coltutricum gliosporids as well as some of the few few species of Fusarium also. Now we are actually working with, in collaboration with Dr. Magesh Kumar, to find out in all what are the symptoms the phytoplasma is associated, and to differentiate which is the symptoms caused by the fungi and phytoplasma. So that work is going on now. And coming to the gaps in our research, so that's etiology and management of emerging disease. We have to find out. Then ecology, biology, and survival of pathogen. Less effort to integrate all the strategies, and the effect of soil nutrition on pathogen, and large-scale field demonstrations to validate the efficacy. So our way forward will be addressing the emerging diseases I have mentioned with cassava, elephant foot yam, and taro. These problems has to be addressed, and integration of various strategies. We are doing different aspects. So all has to be integrated, and development of decision supporting system for the major fungal disease of tuber crop, and studies on effect on nutrition on pathogen suppression. We have not studied much on the role of soil nutrition in soil for the pathogen suppression. This point has to be addressed with the high priority. And another one, investigate soil properties. So also to establish the bioagents, what are the soil properties it's required. This also has to be studied. Thank you.
Thank you, Dr. Veena, for giving a general picture of the disease of tubers. I got two observations. One is your fusarium management of fusarium using lime, 150 grams of lime per plant.
Sir, actually that is for the pH correction, sir.
So that should be again general recommendation. You cannot give that because
no sir, I didn't actually fully. Wherever the pH is between 4 and 5, we have to give the lime application.
That's one thing. Second one is about the fungicides. I think during the May 2020, the government has banned several fungicides, in which both mancozeb and carbendazim comes.
Yes, sir. Now we have to refine our strategies.
Yes.
We have to refine, and we will be giving more importance to bio-intensive strategies.
Yes, because these two, when I give it general thing, because May it was banned during May.
Yes, so we will be hereafter we will be giving, even now also we were giving more importance to biointensive management. So because banning these fungicides, we are left with nothing. So we have to refine our strategies.
I know the problems, but that's whenever the government gives a recommendation like that, that will be a problem. So any other questions from others? Dr. Anagappan?
Sir, Dr. Veena has given a comprehensive presentation. As you suggested that the certain chemicals has been already banned by the government of India, so they have to look for the future strategies to overcome such hurdles. So I think we can move on to the next presentation, and Chairman can request all the speakers to stick on to the time, because we are running short of time and we have so many presentations.
So I think we restrict about 6 to 7 minutes.
Yeah.
Is it okay?
5 minutes, then 1 minute for discussion. 5 plus 1.
Then please, our rapporteurs, will you please ring a bell or something? Show a hand so that they can 1 minute before the time.
Okay, sir.
Yeah.
So next presenter, please. Who is the next one?
Next presentation is by Dr. Amruta.
Yeah.
CTCRA.
Yes, sir. I'm ready, sir.
Please.
Sir, whether my slides are visible?
Yes, visible. Yes, clear.
Okay, sir.
Please, Dr. Amruta, please stick to your time. You have been given with 6 minutes, that is 5 minutes for your presentation and 1 minute for your discussion. Okay, please stick on to the time.
Okay, sir. Thank you, sir. Good morning to all. I'm Amrita. I want to share some of my findings on work characterization of potential endophytes restraining Colletotrichum gliosporoides causing great IAM and diagnosis. We all know that root and tuber crops are fundamental to diets of millions of people. Dioscorea alata, that is our experimental plant, inhabit tropical and subtropical regions and has got wide application in the field of human consumption and medication. The major constraint for the production of this great IAM, that is field disease anthracnose caused by Colletotrichum gliosporoides, resulting in an yield loss up to 50%. Symptoms of the disease include black small lesions.
Doctor, please keep your mic little bit away.
Okay, sir. Okay, sir. Sorry, sir. Sorry, sir. Black small lesions, discoloration, and if the disease severes, it may lead to dieback. How the disease spread is through rain and overhead irrigation, and how you can control the disease is use of resistant varieties and fungicides. So here come the research problem. We know that agrochemicals are nowadays available, improves agriculture production as well as they will manage the disease, but their effect on off-target species is very bad. So here comes the term biointensive approach. It is very important. So here we have used certain endophytes and rhizosphere bacteria to manage anthracnose disease. We know that there is an increased demand for this great IAM, local as well as overseas. So successful control of the disease will promote its widespread cultivation as well as increases total production. So this is experimental plant Dioscorea alata. You can see the Dioscorea alata plant with anthracnose infection in it. Coming to the methodology, we revived potential endophytes as well as rhizosphere bacteria taken from the microbial repository of CTCRI, tested an antagonistic activity against Colletotrichum gliosporoides, screened for phosphate solubilization, nitrogen fixation, potassium solubilization, IAA production, and we done a pot assay in cowpea seeds, and effect of antagonistic endophyte on expression of pathogenicity gene of Colletotrichum gliosporoides, that is CAP20, RAC1 and PAC-C-Vetan. These are the list of endophytes used in this study. Bacillus subtilis, Bacillus pumilus, Bacillus cereus, Bacillus amyloliquefaciens, 2 isolates BAA and BAS, Bacillus licheniformis and Pinabacillus alvae. Here these Bacillus subtilis pumilus, Pinabacillus alvae and Bacillus amyloliquefaciens S, they are identified against Phytophthora colocaceae, but we want to check whether they are showing any inhibitory effect on Colletotrichum gliosporoides also. Coming to the rhizospheric bacteria, E2, Coleus T. Mukta 10, they were also used for the study. These are the antagonists that we have used and these are the results of antagonistic activity. And coming to the results, we can see that BAS, BAA and BC, JKBL showed good result. These are the results of phosphate solubilization where BL and BAA showed good result. Coming to nitrogen fixers and potassium solubilizing bacteria, BC and BL showed good result. IAA production, Pinabacillus alvae and Bacillus licheniformis showed good result. Taken all together, we have done a pot assay where leaf length and plant height were studied in cowpea seeds. 2 treatments were there, seed treatment and spraying treatment. And here we could see that compared to the control, the results were much good in Bacillus cereus, Pinabacillus alvae and M10 and JK. Coming to plant height, result is significant in spraying treatment. But seed treatment compared to control, the result was not that much significant. And we done expression study for CAP20, RAC1 and PAC-C gene in Colletotrichum gliosporoides upon endophyte inoculation. These are the results. These are the amplification conditions, and we could able to see that in the control samples there were amplification for CAP20, RAC and PAC-C genes. But coming to a test sample, that is Colletotrichum gliosporoides co-inoculated with endophyte, we won't get any kind of amplification. But PAC-C, they are expressed, you can see light bands there. These are the results of image analysis by using image A, and you can see that in control they are expressed PAC-C, RAC1 and CAP20. Coming to endophyte co-inoculation, that is test, where you could see that the expression where it's negligible. This has to be further validated by using real-time PCR and all. Let me conclude. Bacillus amyloliquefaciens strains, Bacillus cereus strains, Pinabacillus alvae and the rhizospheric bacteria strains JK and M10. They are good candidates in terms of antagonism against Colletotrichum gliosporoides as well as growth promotion that we obtained from pot assay results as well as growth promotion studies. And noticeable suppression of CAP20, RAC1 and PAC-C where therein Colletotrichum gliosporoides co-inoculated with the endophyte. So these are the future aspects of my study. Thank you.
Thank you, Amrita. Any questions from? Any questions?
Sir, my name is Soumya and I'm from Rubber Research Institute. Sir, I want to ask. Hello, Amrita ma'am, can you hear me?
Yes. No, I'm not ma'am. I'm still doing PhD.
Okay. Amrita, can you show the slides that showing dual culture technique?
Yeah, sure. Sorry, 1 second. Okay. I hope you can see that.
Okay, the first one, BAA, which is that?
Bacillus amyloliquefaciens.
What's the effective range of that inhibition?
You mean about the percentage of inhibition?
Percentage of inhibition.
I think it's more than 80%. I have only shown those isolates that shown more than 80% of inhibition against Gliosporides only here. These all isolates show more than 80% of inhibition.
Okay, thank you so much.
Welcome.
Thank you, Amrita. Then we pass on to the next. We pass on to the next. Who is the next?
The next presentation is by Dr. Sajina from Kerala Agriculture University.
Yes, sir.
Yes. Sajina, please.
Yes, sir. Screen sharing. Share it. Share it. You. Screen share.
Yeah.
Am I audible, sir?
Yes, audible. Please go.
Yeah.
Respected chairman, co-chairman, other dignitaries, and my dear friends, I invite you all to my presentation on rice chaff glycosidia leaf mixture, a promising substrate for the mass multiplication of Trichodoma asperellum. I'm working at Integrated Farming System Research Station. In IFS, we integrate agriculture with allied activities. And for example, if we are taking rice-based IFS, we have the rice cultivation. Along with that, we have the dairy, duck unit, as well as fish culture. And the availability of straw makes the possibility for mushroom cultivation, which is an income-generating activity. But as part of the rice cultivation, we generate the waste material called the rice chaff, which is free of cost, and so it can be used for some beneficial purpose. In Kerala, Gliricidia sepium is a particular plant which is commonly used as a live fence, also as a fodder, green manure crop, and intercrop. It can tolerate repeated cuts. So these leaves can also be further used for some beneficial purpose. And as we know, pesticides are the most commonly used tool for disease management, but they have their own disadvantages, and it is a costly affair to produce the pesticides. So the best alternative is the biological control, which is effective, safe, eco-friendly, and cost-effective. Among the various biocontrol agents, Trichodoma is the wonder fungi, and they have different mode of action which make them strong biocontrol agents. So Kerala Agriculture University has developed a talc-based formulation of Trichodoma asperellum, which is multiplied in cow dung and neem cake in 9:1 ratio for soil application. But this substrate has several disadvantages. Both of them are costly, and neem cake is of low quality and having high EC, so the multiplication of Trichodoma is being affected. And hence the present study was taken up for the evaluation of different substrates for the multiplication and to standardize the quantity and the time of application. So these are the commonly available substrates which are used in different combinations with each other. So we can see the growth of Trichodoma on these different substrates. The first one, cow dung plus neem cake, is the standard one, and the last one we can see rice chaff and the Gliricidia leaf mixture also had better production of the conidia. This is in vivo multiplication also on the substrates under in vivo conditions, and nutrient analysis was conducted before and after multiplication under in vivo conditions. We can see that there was an increase in the nitrogen content after multiplication in 4 media. The phosphorus content was increased in rice chaff plus Gliricidia mixture, and potash also after in 4 substrates. So if we add rice chaff and Gliricidia multiplied with Trichodoma, there is an increase in the content of phosphorus and potash, which is beneficial. And the colony forming units per gram of the substrate was also analyzed, and we can see that the maximum colony forming unit was observed in rice chaff plus Gliricidia as well as in rice chaff plus spent mushroom. And a pot culture study was conducted in amaranth variety Arun for standardizing the quantity and time of application. This is the layout, and the best treatment was rice chaff plus glyrosylia leaf mixture. 250, 500 and 1000 grams of the multiplied media was applied, and as standard control, cow dung plus neem cake was used. So coming to the growth parameters, the plants applied with multiplied trichoderma in rice chaff and glyrosylia as well as cow dung and neem cake at 1 kg per plant at 1 week before planting had the maximum leaf number, leaf area as well as the plant weight. But coming to the root parameters, the treatments didn't significantly differ between root weight and width, but there was difference in the root depth. And incidence of leaf spot and white rust was observed. The leaf spot incidence was non-significant, it was very mild, but white rust incidence was significant among the different treatments. So coming to the benefit cost ratio, if you want to recommend for the farmers, the most beneficial was found to be the trichoderma multiplied in rice chaff plus glyrosylia with a BC ratio of 2.39 followed by cow dung plus neem cake. And this is the harvest of the leaves. We could see the maximum leaf area with a good crop. So I conclude by telling that rice chaff plus glyrosylia leaf is the ideal substrate. Application of trichoderma multiplied in the substrate at the rate of 1 kg per plant 1 week before planting is very beneficial, and it also adds nutrients to the soil. And the study signifies that we can